today we're going to be talking about the cell membrane, the structure of the cell membrane, how it functions, how it lets certain molecules in and out of the cell. Okay? All right, let's go ahead and get started. So in our textbook, this is going to be chapter 7, the cell membrane. Okay? So the first thing that we need to talk about is phospholipids, because phospholipids make up most of the cell membrane, the plasma membrane. Phospholipids are said to be amphipathic. So let me just go ahead and spell that for you. Amphipathic. Okay, amphipathic. Because it's a molecule that has both hydrophilic and hydrophobic parts. It has a part that is polar, and it has a part that is nonpolar, okay? And the phosphate head, and I have a better picture of what the phosphate head looks like in terms of the atoms that make it up, but the phosphate head right over here, this part is hydrophilic, whereas the two tails of our phospholipid is made up of fatty acids, and it's going to be nonpolar. Okay, so it's an amphipathic molecule because it has a hydrophilic or polar side and then a nonpolar side. And as a result, the cell membrane is a bilayer, 2 layers, because the heads can interact with water that's outside of our cells, and then also the heads in the second layer can interact with the water that's inside of our cells. We have both water outside and inside of our cells, and then the tails are facing each other away from the water. So you have to have 2 layers, a bilayer that makes up a cell membrane or plasma membrane, because of the way that our cells are arranged. Okay, we've got water outside, water inside, so you have to have 2. So this is a nice, on the next slide is a nice, and let me just mute that, is a nice diagram showing you, and I gotta move myself here, I'm gonna move myself up here, okay, of phospholipid. So the phospholipid has a phosphate functional group, okay, and a phosphate functional group has a phosphorus atom surrounded by oxygen atoms. And we know that oxygen is electronegative, right? That's a term that you should know by now and you should remember for the rest of the year, okay? It's got a lot of electronegativity, so when oxygen shares electrons with other atoms, it has a stronger pull for the electrons, and because electrons are negatively charged, those oxygen atoms are going to have a negative charge. Okay, so it's this phosphate group that is part of the phospholipid head that makes it hydrophilic, whereas the tails are hydrocarbons. Okay, they're hydrocarbons, and as you guys remember, hydrocarbons are nonpolar, there's no charge, and so the way that a cell membrane looks, okay, you've got the 2 layers because you've got water on the inside of the cell, water on the outside of the cell, and the tails, the nonpolar tails, are facing each other away from the water. Okay, so that's the phospholipid bilayer cell membrane, which is primarily made up of phospholipids. But there are substances that are polar, that are charged, that need to get in the cell. Okay, and we're going to be talking about that in just a minute, but just a little bit more talking about the phospholipid bilayer. So when we look at the phospholipid bilayer, the biggest thickness, okay, the biggest chunk of the cell membrane is nonpolar, right? Because of those hydrophobic tails. So you have this big, thick chunk of the cell membrane that is nonpolar. It's impermeable, meaning it will not let polar molecules in. It's impermeable. It's a barrier, okay? So it's really important that you understand that the cell membrane, the structure, which is mostly phospholipids as a bilayer, the structure does not allow for polar substances to go through. And the reason why is because the biggest barrier, the biggest, thickest part of the cell membrane is the tails that are nonpolar, okay? The nonpolar hydrophobic tails. But sugar's got to get into the cell. Water has to get into the cell. We have to have salts that get into the cell. Remember sugars, like for example, C6H12O6, which is glucose, H2O, which is 2 hydrogens, 1 oxygen. Salt, take for example NaCl. You've got the Na plus ion, Cl minus ion. They need to get in the cell. How do they get in the cell then? That's what we're going to be talking about very soon, in just a few minutes, okay? So the cell membrane, the phospholipid bilayer, it defines the cell. It separates the external environment from the internal environment, okay? And that's actually really important because, you know, in order for the cell to function, have chemical reactions happening inside the cell, it has to be separated from the external environment. It controls what comes in or out. So what substances can cross more easily than others? We just said how the phospholipid bilayer is impermeable to polar substances, so it's only going to let hydrophobic, non-polar substances in easily, slip through the phospholipid bilayer, and also the molecules have to be small, okay? We're going to be talking about how polar molecules get in, how big molecules can get in. Also, I wanted to say that, you know, when we say membrane, the cell membrane, the plasma membrane is what defines the cell, but also you have organelles within the cell, membrane-bound organelles like lysosomes, rough ER, smooth ER, Golgi, mitochondria, chloroplasts, you know, all of those organelles are membrane-bound, meaning that they have a boundary that is made up of a phospholipid bilayer, okay? So the phospholipid bilayer doesn't just pertain to the cell membrane, the plasma membrane that defines the cell, but it also is part of, the phospholipid bilayer is also what makes up the membranes for the organelles, okay? The only organelle that does not have a membrane are ribosomes. Ribosomes are just made up of two things, proteins and RNA, and they are not membrane bound. They're either free floating in the cytosol or they are attached to the rough ER. Okay, so let's move on. And so back in the 1970s, there were a couple of scientists that discovered that it's not just phospholipids that make up the cell membrane, but there's also proteins embedded in the cell membrane. And these proteins can actually move around in the cell membrane. They can move around laterally, forward, back, left, right. And they described it as the fluid mosaic model. Fluid because the molecules, like the phospholipids and the proteins, can move laterally, not up and down, but can move laterally. But also mosaic because the membrane is not just made up of one thing, but it's like a pizza, okay? It's made up of proteins and phospholipids and cholesterol, and we're going to be talking about glycolipids and glycoproteins. Okay, you've got some carbohydrates in there as well. So, as I was saying, how do polar substances get in? Because we need sugar, right? Sugar is what's going to fuel cellular respiration for our mitochondria to make most of the ATP for our cells, right? So membrane becomes semi-permeable to polar molecules via protein channels, okay? And in this example, like water, how does water get in? Through this channel protein or protein channel. How does this amino acid get in? Through this protein channel. How do sugars get in? Through this protein channel, okay? And you know, water actually goes in and out of cells, and they go through these protein channels, and these protein channels are called aquaporins. Aqua, that's supposed to be a Q, porins, and that kind of sounds like what it does. Aqua, like agua or water, porins, like a pore, okay? And these channels are always, they remain open, okay? They remain open, so water can go freely in or out of the cell. And then sugars and amino acids, because they are charged substances or polar substances, they have to be transported through specific channel proteins, okay? That it's sort of like when you get into a line, okay? These sugar molecules, they can only go through one line. They can only come in through the sugar transport proteins. The amino acids can only come in through the amino acid specific proteins, okay? And when I say proteins, you know, these proteins are going to be either in tertiary or quaternary structure, okay? And you can kind of see that behind my face here, okay? But you have the alpha helix, and then it's folded even further. So these cylinders that represent proteins are actually amino acid chains that are in secondary and tertiary structure, okay? So this phospholipid bilayer, right, the biggest chunk, that phospholipid bilayer is nonpolar. Polar substances can't come through, okay? Nonpolar substances that are pretty small, like these guys, can come through, like CO2, O2. Even though they do have oxygen, they're nonpolar. They're nonpolar because if you think about it, the two oxygens are competing, they're sharing electrons, and they have equal pull, right? So O2 is not a polar molecule, and it's pretty small. It can just slip right through the phospholipid bilayer. Same with benzene. Benzene is a pretty small molecule. It's just C6H6, okay? Small polar molecules, and I talked about how water can't come through the phospholipid bilayer. It's got to go through the aquaporin. So let me just draw. Let's say that that's the aquaporin. Majority of water can go through the aquaporin, but every now and then some water molecules can be slip through the phospholipid bilayer because they're so tiny. Water is a tiny molecule, right? It's just got 2 hydrogen atoms, hydrogen being the smallest, atom on our planet, right? And then oxygen, which is relatively small. So H2O can easily get passed through the phospholipid bilayer, but the majority of water molecules are going to go through the aquaporin. Okay. Same with ethanol. Ethanol is a pretty small molecule. It can also slip through the phospholipid bilayer. But larger molecules like glucose or amino acids, they can't. And you can see the arrow here. They can't. They have to go through a transport protein. That's the only way in. Charged ions also cannot go through because they have a charge on them. They're going to be repelled by the phospholipid tails, the nonpolar regions, okay? So now let's talk about proteins. The proteins, these transport proteins. Why are proteins the perfect molecule to build structure in the cell membrane? What allows for the nature of proteins to be the perfect molecule to be the transfer proteins, to allow things in and out of the cell? We have to talk about amino acids, right? Because remember, proteins are made up of amino acids. And let's take a look at these 9 amino acids. These 9 amino acids are part of the nonpolar amino acids. Remember, all amino acids have the same basic structure. They all have an amine group, right? Functional group. They all have a carboxyl functional group. They all have a central carbon atom. They all have a hydrogen atom. Where they differ, where they differ are the R groups. Okay, the R groups are where they are different. And these are the nonpolar R groups. So when I talk about nonpolar amino acids, we're not talking about the other parts of the amino acids like the amine group carboxyl. We're only talking about the R group. These R groups are nonpolar. And if you look at them, what do they have in common? They're mostly carbons and hydrogens. See that? Carbons and hydrogens. These are hydrocarbons. Hydrocarbons are going to be like a lot of fatty acid chains, right? They're going to be nonpolar, okay, nonpolar. I do see some elements here that are not hydrocarbons. However, they are surrounded by hydrocarbons, and so they kind of get lost in there. So that's why it's overall, it's going to be a nonpolar R group. So what about these amino acids, right? If they're nonpolar, in the phospholipid bilayer, you're going to have proteins that are made up of nonpolar amino acids. These guys are going to be the ones that are found in the middle of the phospholipid bilayer interacting with the tails, okay? And I'll point that out when I show you a picture of a protein embedded in the phospholipid bilayer. So these guys are going to be the nonpolar or hydrophobic amino acids. And then you have other ones that are polar, right? These guys are polar. What do these have in common, right? They have electronegative. They have electronegative atoms in the R groups, giving them a charge, okay? They're going to be polar. And then you have electrically charged. Specifically, the acidic ones are going to have a carboxyl functional group at the end. And the reason why they're called acidic is because carboxyl groups, remember the carboxyl group looks like this, right? Well, this is the ionized version of the carboxyl group where the hydrogen atom is lost, Okay, H plus is released into a solution, and by definition, acids increase the amount of H plus in a solution. Therefore, that carboxyl group, okay, maybe I'll draw here, has lost the H plus, releasing the H plus into the solution, increasing the H plus concentration in the solution, making it more acidic. So these two amino acids are considered to be the acidic amino acids because they release their H plus into the solution. It's lost from the carboxyl. Whereas these three amino acids are considered basic because of the amine. They have amine groups here, okay? And these amine groups, the nitrogen can attract the H plus, okay? So in this case, this is the amine group, NH2. It's going to take in a hydrogen atom. It's going to take in an H plus, removing H plus from a solution. That's what bases do, okay? Bases are going to lower the amount of H plus in a solution. But in this case, they're doing it by taking it in and removing it from a solution, okay? So we have all different kinds of amino acids. We've got nonpolar, polar, charged amino acids, and because you have a variety, a lot of different types of amino acids, they're going to be perfect to make proteins that are embedded in the phospholipid bilayer, because the phospholipid bilayer, you've got the middle part of the phospholipid bilayer that is nonpolar, okay? And then the outside is going to be where water is. The inside of the cell is going to be where water is. And then the polar amino acids are going to be on the inside and outside. Okay? And this is our little penguin who says, I like the polar ones the best because penguins live in the South Pole. Get it? Okay, so within the membrane, all right, and let me just highlight this part. Within the membrane, this part of the protein is going to be nonpolar because that's the part that's interacting with the tails, the phospholipid tails. Whereas the ones that are polar parts of the protein are sticking out of the cell and in the cell where there's water, right? There's water outside the cell, there's water inside the cell. So this really, the hydrophobic part, it kind of anchors the protein in there because like, if I took my finger and I like pushed the protein down into the cell, it's going to pop back up because the nonpolar amino acids, they don't want to interact with the water inside, so they're going to go back up. Okay. All right, so now let's talk about like the many functions of membrane proteins. Besides being a channel protein or a transporter protein that we just talked about, these proteins that are embedded in the phospholipid bilayer, they have other functions, okay? Some proteins can act as enzymes. They speed up chemical reactions. So this is what we call a reactant or a substrate. It binds to our enzyme protein and then it comes out as a product, okay? So that's one role or function of a protein that's embedded in the phospholipid bilayer. And then another very important topic that you'll be learning about this year when it comes to cell communication is that you have these signal molecules, and these signal molecules can also be called ligands. Okay, ligands. Ligands can bind to these membrane proteins and change shape, okay, and intracellularly, like inside the cell, it changes shape, the protein changes shape, and it ends up sending a message inside the cell. There's a message that gets inside the cell because of the protein changes shape due to a ligand binding to this membrane protein. And this membrane protein, we call it a receptor. It receives a ligand and it sends the message into the cell, okay? Nothing's actually coming into the cell. Like nothing's coming in. It's just that on the outside of the protein on the outside, it receives a message and then it changes shape. And because it changes shape, it causes this chain reaction of events inside of the cell. Okay, more on that when we go over that later on this year. And then it got kind of cut off in the screen here, but it says cell surface identity marker. Okay, so this protein right here is connected to a carbohydrate chain. Excuse me, a carbohydrate chain, or sometimes we call them oligosaccharides. Okay, oligosaccharides. You know, these are kind of, these are polysaccharides. Okay, long chains, but it's limited to the length, okay? And so you have a shorter length of carbohydrates attached to either a protein or a phospholipid, and they're going to serve as ID markers for the cell, okay? You know, we all have unique antigens. Okay, this is what we call antigens because they are ID markers for the cell. We all have unique ID carbohydrates on the outside of our cells that tell our immune system that this is your cell. And these carbohydrates distinguish your own cells from other cells, from foreign cells, like maybe bacteria that don't belong in your body. So really, these ID markers, carbohydrates, are going to be important for your immune system and identifying cells. Okay, so we've got the glycoproteins. This one is called the glycoprotein. Glyco referring to the glucose, the carbohydrates attached to the protein. This one right here is what we call a glycolipid. Okay, glycolipid, because it's a bunch of glucoses, a chain of glucose attached to the phospholipid. Okay, and then now we have these two proteins that are gluing together two adjacent cells. So if you have two cells that are side by side, they're going to be glued together by these proteins. Okay, and you kind of can't see it here, but there are some proteins embedded in the plasma membrane that are attached to the cytoskeleton, and these remember cytoskeleton is the microtubules, actin filaments, the intermediate filaments, and they help give your cells structure, right, and support. So attached to the proteins are the cytoskeletons that help give the cell its shape, okay? All right, so really the membrane is a collage of proteins and other molecules. So you've got the transport protein, like here, okay? And then you also have cholesterol, these lipids. You know, these lipids are actually cholesterol lipids. They're made in the smooth ER, right? Smooth ER makes lipids, and then they end up being part of the cell membrane. Cholesterol allows for the cell membrane to have a bit more rigidity, and it's so that it's not so fluid, okay? And then you've got the glycolipid, glycoprotein, all those phospholipids, okay? And it's a fluid mosaic because all of these molecules can actually move side to side, back and forward, okay? And mosaic because it's made up of so many different parts. Oh, and I forgot to say, before I forget too, like these guys out here, like right here and here, those are also proteins like collagen, elastin, giving you the, allowing for nearby cells to communicate with each other, and also giving tissue structure, okay? But out here, this is called the extracellular matrix, or ECM for short. Extracellular matrix, meaning on the outside of the cell, this matrices. It's made up of proteins and carbohydrates and all sorts of organic things, okay? So I do want to talk a little bit more about those antigens, right? Those ID markers. These membrane carbohydrates, they play a really key role in cell-to-cell recognition. It helps your immune system recognize your cells as being self, your self cells, okay? Ability of a cell to distinguish one cell from another, and these are what we call antigens, okay, antigens. It's important in organ and tissue development, and it's a basis for rejection of foreign cells by the immune system, because if you get infected with certain types of bacteria, your immune system is going to look at those carbohydrates and see that they're not your self cell, okay? Okay, so that's the end of membrane structure. Obviously, I can't take any questions because I'm just talking to myself in this video. Now let's talk about the movement across the cell membrane. How do things get in and out of the cell? This is a pretty amazing computer-generated image. This interior portion right here, these are the fatty acid tails, the hydrophobic tails of the phospholipids, okay? And the orange and green, those are the phospholipid heads. And then the protein, this is a picture of computer generated image, not a picture. Computer generated image of an aquaporin in tertiary structure, right? You could see the alpha helix coiling. And then water molecules are going through the aquaporin, and there's all this water outside. You can see all the water molecules outside, and also, well, you can't see it on the inside, but there's a lot of water on the outside and inside, and there's water going in and out of the cell through the aquaporin. So we need to talk about diffusion. Hold on one sec. Let me just have a quick drink of my coffee. Okay, so when we talk about diffusion, we have to also talk about the second law of thermodynamics. Basically, this is a law that governs throughout our universe, okay? It just states that things go from order to disorder, and that is entropy. Entropy says that things go from order to disorder. And I've mentioned this before when I was teaching this lesson about how when you clean your room, right? When you clean your room, everything's in order, right? But how long does it take before it gets disorganized again? Disorder, right? I clean up my room, and then maybe the next day or maybe in the next week, it gets all disorganized again, right? That's an example of entropy. So if, for example, you have order in that you have a high concentration of molecules on one side, well, diffusion, because of entropy, those molecules are going to move and scatter. They're going to move and scatter, and they're going to spread out until equilibrium is reached, okay? You know, equilibrium means like equal, right? So it's equilibrium is when all the molecules are just spread out and they're moving around and just spread out. We went from order to disorder, scattered, okay? So diffusion is movement from high to low concentrations. Diffusion is movement of molecules from high to low concentration. And do you need to input any energy to do this? No, this is a natural law in our universe, okay? You don't have to input energy. If I spray some perfume in my room, the perfume molecules are gonna scatter. It's gonna dissipate eventually, okay? I didn't need to do anything except, pump the perfume, but the molecules just spread out on their own. It doesn't require energy, okay? So simple diffusion is when molecules move from high to low concentration. It's what we call passive transport. Passive because there's no energy needed. It just happens on its own, okay? There's no energy needed until equilibrium is reached. Equilibrium. All right, but then there's also something called facilitated diffusion, okay? Facilitated diffusion. This is movement of molecules from high concentration to low, but if it's a polar molecule, it cannot go through the phospholipid bilayer alone, through the phospholipids. It has to go through a transport protein, okay, or membrane protein. So if I have a lot of molecules on this side of the membrane, they're gonna be facilitated or helped to go from this high concentration to a lower concentration area through a transport protein. So the open channel, it's pretty fast. These molecules are moving through pretty fast from high to low. There's no energy needed here because this is an example of diffusion, but it is specifically facilitated diffusion because these molecules are polar. Okay, because these molecules are polar, they have to go through a transport protein. Okay, whereas active transport is kind of like what it sounds like. Active, it requires energy. Okay, it requires energy because you're actually moving molecules against the concentration gradient. Okay, I probably should have gone over concentration gradient on the previous slide, so let me do that really quick, okay? If I have a lot of solutes, right? Solutes are substances dissolved in a solvent, okay? So if I have a lot of solute dissolved on this side of the membrane versus this side of the membrane, maybe this side of the membrane I only have a few, but there's a lot of sugar or whatever solutes dissolved outside my cell and not as much inside the cell. That's what we call a concentration gradient, okay? Concentration gradient. a gradient, right, is where there's a difference, right? A difference across a membrane. There's a high concentration on the outside versus the inside. That's what we mean by concentration gradient. There's a difference in concentration between two environments or two areas across a cell membrane or across a membrane. And then, the higher the difference, there's a higher concentration gradient. So active transport is where cells, they're going to move molecules against the concentration gradient. In other words, they are moving molecules from a low concentration to a high concentration. Okay, I kind of don't like this picture because I don't think it really shows you what's going on. But, if we have, again, a high concentration of solutes outside the cell here, okay, and then there's just a few here. Active transport is when a protein is going to pump the solute outside where it's already crowded. It's going against the flow, against the concentration gradient, okay? So this protein, we call it a pump because it requires energy to pump, and it's pumping it against the concentration gradient. Think of it this way, like think of a protein pump or active transport as like you entering an already crowded room. It's already crowded, packed with people. You got to push your way through, okay? So active transport is sort of like that in that it's pushing molecules against the concentration gradient, not going down their concentration, but going the opposite, and it's going to require ATP. And whenever you guys see that word pump in regards to a protein, you know that it's active transport because it's active transport because it requires energy to pump against the concentration gradient. Okay, so an example of this is supposed to be a GIF. Oh, it's working. Okay. It's a GIF that shows you the sodium-potassium pump. The sodium-potassium pump is a protein that's found in all of your cells. Every single one of your cells has a bunch of these guys, okay? And using ATP, they actively pump out, okay, 3 sodium ions at the same time, and then they actively pump in 2 potassium ions inside. And so you can see the 3 sodium ions going out, and then these 2 potassiums coming in. And it creates a concentration gradient, okay? All of your cells, so let me just draw as if our cells were like a sphere. We have a high concentration of sodium ions outside, and then a high concentration of potassium ions inside, okay? And the sodium-potassium pump maintains this concentration gradient. And it maintains this concentration gradient for a lot of different reasons, but a really good example of why your cells, especially in your nervous system, your neurons need to maintain the sodium-potassium pump in order for the neuron to create what we call an action potential, which is the mechanism for a neuron to send a message to another neuron. So when your brain cells communicate with other brain cells, an action potential has to move through these neurons. And the only way it can do that is by having first the sodium-potassium pump create a concentration gradient, okay? And what's happening in this picture, and it's not very clear, I think that this GIF is not that accurate, in that what happens is that ATP, which is adenosine triphosphate, it loses one phosphate group and becomes ADP. The phosphate group, an inorganic phosphate group, what it does, it attaches itself to the protein and it energizes that protein. So it's really the inorganic phosphate that attaches itself to the protein, energizes the protein. I feel like in this GIF it shows it looks like it's ADP that's attaching itself to the protein, but it's not. It's an inorganic phosphate. So just a really quick lesson on ATP. So ATP is a nucleoside, right? It's a type of nucleotide, adenine. You've got the sugar. This is our 5-carbon sugar. You've got the adenine, nitrogenous base, and then you also have 3 phosphate groups, okay? And these phosphate groups, sorry, it's so messy. But these phosphate groups, there's 3 of them, triphosphate. There's covalent bonds that hold the phosphate groups together. They are there's energy stored in those covalent bonds. And when this third phosphate group, the bond between the second and third phosphate group is broken, there's a release of energy. This phosphate group, this last one, it carries the energy with it. It's going to attach itself to this protein, energizing that protein. Okay? All right, so that's how ATP energizes this pump, this protein pump, the sodium potassium pump. Okay, so as a really quick review, passive transport, no energy required. Okay, so you need to remember things about passive transport. No ATP required, right? And also the movement of molecules are going to go from high to low. Okay, simple diffusion, right? That doesn't require a protein. Whereas facilitated transport or facilitated diffusion, it does require a protein. That's the only difference, okay? So you guys, that's the only difference between that's the only difference between simple diffusion and facilitated diffusion. Facilitated transport diffusion is facilitated by, helped with the protein, okay? So simple diffusion, nonpolar, hydrophobic, small, I should add small in there, molecules can simply diffuse through the phospholipid bilayer. Whereas facilitated diffusion using a protein, you're dealing with polar, hydrophilic molecules that cannot come simply through the phospholipid bilayer. They have to go through a protein channel. Active transport, yes, requires ATP. ATP needed, okay? And then it's not from high to low, but the protein pump is pumping molecules from low to high. It's creating a concentration gradient, right? It's creating this concentration gradient where there's more on one side of the membrane, okay? And so it's a protein pump which requires ATP. So I like this picture too. Simple diffusion goes right through the phospholipid bilayer. It's got to be non-polar. It's got to be small, okay? Whereas over here, polar, and they're polar charged, so they're going to have to move down their concentration gradient through the protein. Whereas active transport, it doesn't necessarily have to be non-polar polar, but it's like in the case of the sodium potassium pump, it's a charged ion that's going to be pumped against the concentration gradient into an area where it's already crowded. It requires ATP. Okay, let's say that the molecules are like super large. Okay, if it's super large, they can't fit through a protein. So what happens is that the cell membrane will change shape. It's going to either, if it's bringing something in, it's going to wrap around the substance that needs to come in, or if it's something that needs to leave the cell, there's a vesicle that's going to release or secrete the substance. So endo means in. Endocytosis is where you have cells bringing in large substances. Like phagocytosis is like solid particles. Pinocytosis is like cell drinking, where there's a lot of solutes dissolved in a solvent that have to come in. Exocytosis is where the cell is secreting something that's made inside the cell. Like in this picture, and let me just, I think you're gonna have to move me here. In this picture, this is a vesicle that contains, like, let's say proteins that were made on the rough ER. These proteins need to be secreted. So the vesicle, the cell membrane and the vesicle membrane are gonna fuse together and they become one. And it releases all of those proteins into the extracellular matrix, wherever it needs to go, okay? And that's called exocytosis because it's going outside, okay? All right, and then the next one. So yeah, phagocytosis is coming in, the solid particle. Pinocytosis is dissolved substances, solutes coming in. And then there's also cell receptor-mediated endocytosis. See the cell membrane here? The cell membrane has these receptor proteins that bind to these signals that are floating that are part of the solute, okay, on the outside. And once they attach to the receptor protein, it causes the cell to respond in such a way that the cell membrane folds inward and forms a vesicle and bringing in the substance, okay? So I think that, yeah, this is the last topic from our video is osmosis. Okay, water is very important to life, so we have to talk about water separately. But basically, guys, osmosis is diffusion of water, facilitated diffusion of water. That's what osmosis is. And water is going from where there's a lot of water to an area where there's less water. And when we talk about more water, less water, that's in regards to the solute level. Okay, solutes, remember, are the stuff that's dissolved in the water. If you have a lot of solute, something that's really I was going to say hypertonic, but I haven't introduced that word yet on the PowerPoint. But if there's a lot of solute, then there's going to be less water. And then if it's very dilute, where there's less solute, then it's going to be more water, right? I think I will go back to this YouTube. Okay, it's not YouTube like you're watching this on YouTube, but it's a U-tube because it's a U-shaped tube. I'm going to come back to this. So water, okay, is always going to go from an area or a solution where there's less solute, more water, to an area where there's more solute, less water. So the direction of osmosis is determined by comparing total solute concentration. So when I compare the solution here on the left side, so this is my left side, this is my right side, I've got more water on the left side, okay? And I've got less solute on the left side. So by definition, this solution is hypotonic. On the right side, this solution is hypertonic. You guys, these terms, we only use these terms when we are comparing, okay? When we're comparing solutions, that's when we use these terms, okay? You cannot use these terms singularly. When something's hypertonic, that means there's a lot of solutes in it compared to something else, another solution, okay? So on the left here, we've got a hypotonic solution. On the right, hyper. One way I remember what hypertonic means is like hypertonic, like there's a lot of sugar, a lot of solute. When you drink something that's really hypertonic, like a lot of sugar, it gets you hyper, okay? So water, by osmosis, it's going to flow from an area where it's hypotonic to an area where it's hypertonic. And it's basically trying to reach equilibrium, okay? This is a type of diffusion, right? Doesn't require energy. It just happens naturally, where water is going to go from where there's a lot of water to less water by diffusion. So it goes from hypotonic to hypertonic. And then isotonic, that prefix iso means equal. When you compare these two solutions and they're isotonic to each other, that means they have an equal amount of water and an equal amount of solute. So I'm going to go back to this U-shaped tube. And this U-shaped tube has a semi-permeable membrane, okay? There's a membrane that's semi-permeable. Just like how our cell membrane, plasma membrane, is semi-permeable, you could also have this U-tube with a semi-permeable membrane, and there's basically little pores. There's little holes and pores in the semi-permeable membrane that only allows for small, very small molecules to go past, okay? So these larger molecules, like these purple dye particles, they can't go through because they're too big. The only thing that can go through is water, okay? On the left side, I see that there's less solute, more water. On the right side, I see there's more solute. And so we describe the left side as being hypotonic, right side, lots of solute, hypertonic. So then water is going to flow. Because water is so tiny, water is going to flow through the pores to the other side. And as a result, this side of the column of the U-tube is going to rise up. This side is going to go down, okay? Because water is flowing. And this happens naturally, okay? There's no energy required for osmosis. So let's take a look at an animal cell in a hypotonic solution. I'm going to just redraw this really quickly. So if I've got a hypotonic solution out here, that means, remember we use these terms in comparison, that means this animal cell is hypertonic, right? If it's hypo outside, that means the cell itself is hypertonic. I mean, our cells, we have a lot of stuff inside of our cells. Besides all of the organelles, we've got a lot of different molecules and solutes within our cells. So hypotonic, on the outside, maybe just a few, okay? So then by osmosis, which way, which direction is the water going to go? Water is going to go inside the cell, right? Because water will go from hypotonic to hypertonic. Water is going to go in. And if there's a huge concentration gradient, if there's a big difference between the outside and the inside, then it's very possible that the cell, the animal cell, is going to lyse. It's going to burst open. Okay, that's what's happening in this picture. It's like a water balloon that quickly fills up with water and then it just bursts open because there's so much water coming in. That's animal cells. But in a hypotonic solution, plant cells do really well. Plant cells become turgid. Okay, there's something called turgor pressure. Because as the water comes into the plant cell, you know, the vacuole fills up, it swells up, the cell swells up, and the cell is swollen, and it actually enables the plant body, the plant itself, to stand upright. Okay? You know, I always ask my students, I said, how come the animal cell bursts and lyses, but why does the plant cell not burst and lyse? Plant cells also have a cell membrane, right? Well, in addition to a cell membrane, plants also have a cell wall. This cell wall is made out of cellulose, right? And cellulose is a very tough polysaccharide. It's a very rigid, strong polysaccharide. The cell wall is made out of cellulose. So it's what keeps the cell from bursting. Okay, it's what keeps the cell from bursting. So turgid or turgor pressure is this internal pressure, water pressure, where water's coming in and it's pressing on the cell wall. Okay, and it allows for the plant cell to swell up but not burst. And it enables the whole plant itself to stand upright, okay? However, in an isotonic solution, let's talk about a plant cell first. In an isotonic solution, right, there's equal amount of solute inside or out, so there's equal amount of water going in and out. And for a plant cell, it's going to become flaccid, okay? Flaccid means it's like, it's limp and it's bending over, okay? It's, yeah, so a plant is no longer standing upright, but it's lost, you know, its uprightness and it's bent over, okay? And that's not good, right? Because when plants are bent over, then they don't get maximum sun exposure for photosynthesis, right? Plants want to be upright and get as much sun exposure as possible. So a hypotonic solution would be the most best possible environment for plants, okay? Whereas isotonic solution is the best possible environment for animal cells, because animal cells in isotonic solution, you have water coming in, water going out. There's no lysing, okay? It's at its normal state. However, in a hypertonic solution where there's a lot of solute on the outside, okay? If there's a lot of solutes on the outside, then water is going to come out. And as a result, the animal cell is going to shrivel up. Okay, it's going to shrivel up. Plant cells are also going to shrivel up. We call that plasmolyze because they have a cell wall that helps maintain the shape of the cell, but it's a cell membrane that kind of just shrivels up. And plants can actually come back from this. If they don't stay plasmolyzed for a long period of time, you can actually revive plants. Okay, I mean, it's sort of like, you know, when a plant is kind of has lost water and it's really, you know, leaning down for a long time, it can plasmolyze, but then if you just water it, it could just come back right up, okay? So in a hypertonic solution, animal cells are going to shrivel up. So in a hypotonic solution, a cell in like fresh water is an example of a hypotonic solution. We're going to talk about paramecium and the contractile vacuole on the next slide. But plant cells, right, they, you know, ideally need to be in a hypotonic solution, so they're turgid, which is their normal state, where it's full and the cell wall protects them from bursting. So that paramecium that I was talking about, paramecium are single-celled organisms. They're called protists, okay? And the thing about them is they live in freshwater environments. The problem with living in a freshwater environment is that it's hypotonic outside, right? If it's hypotonic outside, that means that the cell itself is hypertonic. So water's coming in, Water is coming into this little body, and when water comes in, it can burst. Well, they have a unique organelle called the contractile vacuole, which is right over here. It's like a water balloon. It fills up with water, and then in this picture down here, it squeezes and pumps and dumps the water out. Okay? And this contractile vacuole in a very hypotonic solution, all this water is coming in, and it's going to quickly fill up, quickly pump the water out, quickly fill up, pump the water out. And it works like this 24/7, okay? 24 hours a day, 7 days a week, okay? And of course, it requires ATP. Okay, in a hypertonic solution, you know, both animal and plant cells are gonna like shrivel up, okay? They can die if they've shriveled up for a long period of time, okay? Yeah, I did say how plants they can wilt, but they'll wilt, but then they can recover from it. Isotonic solution, animal cells are normal in an isotonic solution. Like when you go to the hospital and you are like hooked up to an IV, right? An IV solution is basically water with salt in there. And it's usually, the IV bag will say, where's my pen? The IV bag will say 0.9% NaCl, okay? That's isotonic to your blood. 0.9% NaCl is isotonic to your blood. And so, yeah, that's down here. Slightly salty IV solution in the hospital. And plant cells, they don't prefer to be in isotonic. What's most beneficial to plant cells is being in a hypotonic solution. So I did mention how aquaporins are the proteins that allow for water to go in or out of the cell. That's how osmosis works. Osmosis goes in and out of cells through the aquaporins. Okay, the last thing we're going to talk about is on this slide, right? You'll see questions like this on the AP exam, on our quiz in class, okay? But you're basically comparing the solute concentration of a cell versus the environment. Inside the cell here, okay, it has 0.03 molar sucrose. I'm just kind of rewriting this for some reason. And then 0.02 molar glucose. Those are types of sugars, right? Or saccharides. If you add up the molarity, right? Molarity is just a measurement of concentration. It's 0.05 molar. So that's how many the concentration of solute inside the cell. Whereas on the outside, you've got 0.01, 0.01, 0.01. All together out here, it's going to be 0.03 molar solute concentration. So which one's hypotonic, hypertonic? The cell is going to be what? Compared to the solution outside, the cell is going to be hypertonic, right? And then the beaker, the solution on the outside, is going to be therefore hypotonic, right? We only use these terms when we compare solutions. And so which way is the water going to go? Which way is the water going to go? Water is going to end up going in. Water, okay, H2O. H2O, water is going to end up going into the cell because it's hypotonic out here, where it's hypertonic inside. There's more solute inside, less solute outside. Water is going to move in to reach equilibrium. Can we say that this cell is going to lyse or burst? I can't say, because honestly, I feel like these two molarities, when we compare them, there's not a huge concentration gradient. It's not that much different. If it was a huge gradient, then there's a possibility. Like if the outside was much more hypotonic, then yeah, there is a possibility that that cell would lyse and burst if it was much more hypotonic outside and much more hypertonic on the inside. Okay? So that is the end of our lesson. I hope that this video helped you understand the structure and function of the cell membrane and how things get in and out of the cell. Okay. All right. Have a beautiful day, and I'll talk to you later. Bye.